let everyone into the meeting and then we're going to get started. Thanks for being here today. Welcome on behalf of Great Pond Foundation and the Island Boards of Health to our third annual MB-CYANO data review. Thank you for joining us today. As a reminder, there's a lot of us in this room, so if you could please keep your microphones on mute to start. We'll have time for questions later. For those of you who don't know me, my name's Emily Reddington. I'm a scientist and I'm the director or executive director of Great Pond Foundation. I'm joined here by my colleague, David Bauk, who's the director of science and collaboration and who will really be taking us through the data today. We also have the Island Board of Health agents that we'll bring up later because they're the experts at answering all of our questions about how to stay safe. For those of you who've not attended a prior MB-CYANO presentation, I want to remind you that this program is created on the island by the island community, and it's a collaboration between scientists from Great Pond Foundation and Chilmark Pond Foundation and the Island Boards of Health. Entering 2024, this is our 4th season with the program. Today, our agenda is briefly to go over what are cyanobacteria, where do you find them, and what's the MB-CYANO program. We're then going to jump into the meat of the meeting today, which is going over the data from 2023. What did we learn last year? What were the cyanobacteria doing? What are the data and the trends? And then a few practical tips for the public. How do we understand blooms? What do we do about them? And at the end, we'll have a question and answer. So MB-CYANO is made possible by funding provided by the Edie Foundation, the Chilmark Pond Foundation, participating towns, as well as those through the generous support by individual and foundation donations to Great Pond Foundation. Participants are welcome to type questions into the Q&A section during the chat or into the chat for Q&A. And if we have time, we'll answer it as we go. If not, we'll get to those in order at the end. So during the Q&A session at the end of this presentation, we'll allow participants to raise their hand and invite them to speak, put on their camera, turn on their microphone. We don't control those settings, so we're asking you to kind of keep a hand on those, and thank you so much. If you have any questions that we don't get to during the session, please feel free to email MB-CYANO, that's MB-CYANO, at greatpondfoundation.org, or you can visit our website, greatpondfoundation.org/MB-CYANO, or just put MB-CYANO into your Google browser. Without further ado, please let me introduce GPF's Director of Science and Collaboration, David Bauk.
Hey everyone, my name's David. I'm the Director of Science and Collaboration. Thanks Emily for the intro, and thanks to everyone for being here today and the Island Boards of Health for attending. I will be kind of going through our data as Emily talked about and just kind of reviewing the basics of the MB-CYANO program. So let's get right into it. So first of all, what are cyanobacteria? Put simply, they're a type of photosynthetic aquatic bacteria that occur naturally in both freshwater and marine ecosystems. They're microscopic, but can grow in colonies large enough to see. Most types of cyanobacteria that we encounter are more freshwater-oriented species, and where things can get interesting and where things are actually less well understood are those environments where freshwater and seawater mix. Also known as a brackish environment, and which is a characteristic of many of the ponds on Martha's Vineyard. In general, the peak period of growth for cyanobacteria occurs during the August to late October timeframe, which corresponds more or less with the hottest times of the year. And often we see an acceleration of growth patterns coincide with those increasing ambient temperatures. And a couple other interesting facts about the cyanobacteria branch of the tree of life. Cyanobacteria are actually one of the oldest forms of life on the planet. Some of the oldest fossils found are of cyanobacteria, dating back to approximately 3.5 billion years ago. And life on planet Earth actually has a lot to be thankful with regards to cyanobacteria. The oxygen atmosphere that we depend on today was generated by ancient cyanobacteria growth. And they are also the precursor to modern plants. through a process called endosymbiosis, which is essentially one organism being absorbed by another and then forming a symbiotic relationship. A cyanobacterium exists in the chloroplast of plants, and they are essential for the process of photosynthesis, which we all know is very important. So what is the big deal? Why do we monitor cyanobacteria growth? Well, some genus and species of cyanobacteria are known to produce toxins under the right conditions. When in contact or ingested in concentrated amounts, these compounds can cause adverse health effects that can be severe depending on the level of exposure. An increase in favorable conditions required for cyanobacteria growth have resulted in an increasing frequency of blooms worldwide. So we're not alone in this problem. And this is coupled with an increasing awareness and ever-evolving understanding of their specific biology. So when I say a cyanobacteria bloom, what am I actually talking about? So a bloom is essentially accelerated growth. It is growth that is outside of their kind of normal growth conditions. The conditions are present that allows them to grow rapidly. And that is what we call a bloom. And what contributes to growth is primarily these factors. You have the abundance of nutrients, primarily nitrogen and phosphorus. You have warmer temperatures, which coincides with that summer season. And just like all plants or photosynthetic organisms, the availability of light, which also kind of corresponds with the summertime and the greater light availability. Now, it's actually impossible to know visually if a cyanobacteria bloom is producing toxins. It must be tested by laboratory technicians, which can be a complicated procedure. So what do we do to kind of assess the threat if toxin testing can take a little time or isn't a quick turnaround time or it's not something easy to do? Well, we can perform regular monitoring. And regular monitoring of cyanobacteria density and concentration patterns can help to judge if potentially harmful conditions are present. And so this includes direct measures of concentration in a sample, which can be measured usually within just a few hours of a sample being collected. We can perform microscopic analysis on the same sample to judge if the genus present are known toxin-forming species or genuses. And also regular monitoring of not just the cyanobacteria concentrations, but the environmental conditions present allow us to analyze some of the long-term seasonal trends and patterns in their growth and community structure. And this can this allows us to begin to learn how they interact and function in each particular pond and environment. And really, there still is a lot that we don't actually know. And the greater field of cyanobacteria research is kind of ever evolving and ever changing and always being updated. So it is a learning curve. And here in these pictures, you can see some examples of cyanobacteria blooms. We have one here from 2020 near the Abels Hill parking lot. You can see the green scum kind of forming along the shoreline and out into the main water column. We have this bloom that occurred in 2021 in Squibnocket Pond. This is a great aerial shot of a very widespread bloom that occurred in this area. You can see the diffuse characteristics of that bloom. And then here in 2022, we had another kind of high concentration bloom also in Chilmart Pond area. And you can see the kind of pea soup green consistency and, kind of the thick concentration of it that exists. And these are true examples of a cyanobacteria bloom. So what is MV cyano? During late 2020 and early 2021, in recognition of the emerging need to monitor and understand the local dynamics of cyanobacteria growth in vineyard ponds, MV cyano was developed through a collaboration between scientists from the Great Pond Foundation and Chilmart Pond Foundation, as well as with the Island Boards of Health. Together, this group developed a system of monitoring and risk assessment based on the current body of knowledge and research surrounding cyanobacteria biology and public health. These assessments are based off threshold concentrations and other key metrics as advised by the Environmental Protection Agency, the World Health Organization, Massachusetts Department of Health. And what we have here on the right is kind of what that threat assessment matrix or the advisory matrix looks like. We have a stoplight system going from green to yellow to orange to red of increasing kind of risk thresholds, and with each of these kind of categories that are informed by those key metrics that we're drawing from other sources around the world, we can kind of assess based on the conditions present what activities should be used in caution, and maybe when they should be avoided, depending on the severity of the cyanobacteria activity in that pond or in that location. There are currently no federal or state mandated laws or regulations surrounding cyanobacteria concentrations in recreational waters. So advisories, what you see here, are at the discretion of local boards of health and town officials. So we're about to dive into the actual specific data from 2023. Here's a quick roadmap of what we're going to cover for the bulk of this presentation. First, we're going to review where our cyano monitoring occurs. We're going to take a look at how data is collected, how it's analyzed, what we're actually collecting to make these reports. We'll do a quick overview of how to interpret that data, and then we'll kind of dive into the individual ponds and look at the trends from 2023. We'll cap that up with some recommendations for the public on what to do if you possibly encounter a bloom, things to watch out for as we head into the 2024 summer season, and have some time for questions at the end. So our monitoring locations, so Great Pond Foundation and staff from the Island Boards of Health conduct regular monitoring in 8 ponds. Cyanobacteria samples are collected alongside other water quality monitoring data at multiple stations throughout an ecosystem. For example, we have here in Chilmark Pond, you can see all the blue dots correspond to where we monitor for cyanobacteria and other environmental water quality metrics. Same goes for these ponds as well. Other samples are collected from key interface points of public beaches or ponds, such as the swimming pond or swimming beach at Cess Pond in West Tisbury. And then other ecosystems such as Watcha Pond or Tashmoo, Lagoon, Senjikitaket are monitored. Either they can be monitored regularly or on a needs basis, depending on the conditions. And while we may not conduct regular monitoring everywhere across the island, there are a lot of ponds in Martha's Vineyard. We're always able to investigate a new or potential bloom if one is reported. So how do we analyze the data? So a cyanobacteria sample is simply a surface water sample that's collected at those regular monitoring locations or at the site of a potential bloom. And we use specialized amber or opaque bottles, which limit photosynthetic activity in the sample after collection. And this is in order to preserve the relative concentrations and conditions of the sample until it can be processed shortly thereafter. CYANO utilizes a specialized instrument called a fluoroprobe, this device here, to rapidly measure and estimate cyanobacteria concentrations in a water sample. And cyanobacteria and other classes of microscopic algae, also known as phytoplankton, utilize a pigment called chlorophyll in the process of photosynthesis. And some of these other classes of phytoplankton are green algae, diatoms, and cryptophytes. The fluoroprobe, which is manufactured by BB Malndecki, if I pronounce that right, utilizes the principles of fluorescence, which is essentially the absorption and reflectance of light by those chlorophyll pigments, from different types of phytoplankton to calculate the relative concentrations of each class in a water sample. So each phytoplankton class fluoresces at different wavelengths of light in the spectrum. Broad spectrum LEDs are pulsed through each sample in the fluoroprobe, and that allows for the varying concentrations of each class to be calculated. And so this essentially in a few seconds can provide us with the relative concentration of cyanobacteria in that sample. In samples where we have higher concentrations, microscopic analysis can be performed to try and identify the genus of cyanobacteria in that sample, whether it's of that toxin-forming variety. And in the same cases of high concentration, along with several other determining factors, a sample can be sent to our collaborators at the Gobler Laboratory at Stony Brook University on Long Island for an actual toxin analysis. So now that we kind of understand the what, the why, and the how of the CYANO program, we can take a tour of our monitoring results from last summer and the trends in cyanobacteria growth that we have been observing. Before we look at each specific pond, I thought it'd be good to kind of just review how to interpret our data. So we have here, it's just kind of an example slide, the basic components of a graph. So first of all, up here at the top, you'll always have where the data was collected. So in this case, this is an example from Squibnocket Pond. We have the year of collection. denoted right here with a bit of a legend to show that if we have multiple years kind of next to each other, they'll have different graphics to represent them. The coloration, the shading that you see and behind the graph, that'll be on every single one. That corresponds with that advisory matrix that I pointed out earlier in the presentation. So it corresponds with the relative thresholds where a sample crosses in to that advisory category, either a full bloom advisory, a watch, or the yellow alert status. Down here at the bottom on the x-axis, you will always see a time reference. So this is the date. And so it kind of shows the timeline of the cyanobacteria results throughout the sampling period. And probably most importantly to pay attention to is this y-axis here on this side of the graph. This is your cyanobacteria concentration in micrograms per liter. It's a volume. And this scale will change depending on the range of the data in that pond or in that particular data set. So in this example, we have a pretty even spread from 0 to 125 micrograms per liter. You can see, just barely see all 4 of the color codings. But in other ponds, we might see that scale is much, much higher, depending on where, the concentration of the samples that we saw in that pond. So just keep in mind that these scales are not the same. They will change relative to the data set itself as we progress. All right, so starting in Chilmark. Now, as we go through each of these ponds, I'm first going to introduce the pond itself, tell you a little bit more about the characteristics of that ecosystem, and run through the trends in cyanobacteria measurements. We'll take a look in depth at the 2023 season, and then look at some overall comparisons from different years where we have multi-year data. And then we'll also look at different types of cyanobacteria that were observed in those locations whenever possible. So first up, we have Chilmark Pond. So you can see it's located here on the south side of the island. Here is a map of that pond and that ecosystem with all of our monitoring stations included. And each of these stations has a station name, which corresponds to the graph on the right, which shows the sampling results of each station throughout the entire sampling season. And again, on the bottom, in this kind of column view, you see the timeline here for both of these. And then you can see here on the y-axis, the concentration of cyanobacterium present at each of those stations. And for example, this scale is very high because we had a high reading here at this station last summer. Now, Chilmark Pond is kind of an interesting system. It can kind of be broken into 2 zones. You have the lower pond here on the west, which is primarily a brackish ecosystem. It does have an opening that's manually created on the south side of the pond here intermittently throughout the year that provides seawater, creating those brackish conditions. And then you have kind of the upper pond system over here to the east, which includes these 2 ponds that are connected to the lower pond via these narrow creeks. These are fed by several freshwater streams that flow in, as well as a lot of groundwater influx. And so this area of the pond tends to be much more fresh and creates kind of a freshwater gradient the further you move from west to east. And that is important because a lot of cyanobacteria species tend to be more freshwater oriented. Now, due to the natural topography of the environment in this pond, water does tend to only flow this direction. And this area does tend to be where we see our highest cyanobacteria concentrations and has been kind of the site of previous blooms. So you can see here in our data, looking at the stations in the lower pond, they tend to have lower concentrations throughout the summer. And then it isn't until you kind of get into this more freshwater zone that you start to see higher spikes and blooms of cyanobacteria in these areas. And one interesting thing of note from last year is probably for the first time in our monitoring experience, we actually saw a bloom of cyanobacteria or a high concentration occur at this station over here at Chilmark Pond 4. And that actually was due to, if you look at the time frame, it actually corresponds with the bloom that occurred in the upper pond system. And what we think happened is that the natural currents flowing this way, and then the natural wind patterns kind of actually first dispersed the material into the main lower pond and to the point where we weren't picking it up at those stations, and then reaccumulated on the downwind side of the pond. That's a really important point because the algal material or the actual physical cyanobacterial bloom, while it may not initially propagate in this area due to unfavorable conditions, it is still subject to the motion of the wind, the waves, and the currents, and can move around in a pond if in high enough concentrations and given the right conditions. So that's always something we want to keep an eye out for. And as we can see, we did have a bloom kind of in that same August to October peak growth period last year. We are missing data from this station here at Chilmark Pond Upper, simply because the bloom was kind of extensive throughout this area and we did not want to start splashing around collecting samples. Now, kind of focusing in on that particular bloom, you can see it was kind of the pea soup green consistency, pretty diffuse throughout, and then kind of mapping in areas of low flow or wind consolidation. And this bloom, after looking at our microscopy images, is primarily dominated by this genus Dolichospermum. And this was also confirmed by the Govlu laboratory. Looking at Chilmark Pond from different years, so you can see here we have, again, the scale has changed a bit because we have a higher sample from 2022, so the scale is relative. Also, too, this cyanobacteria concentration is the average across the entire pond, just so we can compare the years easily. So it's the average of all the stations. But the primary, the bulk of the concentration of cyanobacteria is coming from the blooms that were experienced in this upper region of the pond. So the lower pond has always kind of remained low in cyanobacteria, but most of these blooms are occurring in this area. And as we can see, 2021 is denoted by a different color. So you can have that here on the graph. You can just barely see 2021. It was actually a fairly low year for cyanobacteria. We had low concentrations throughout. And then that is contrasted by 2022, and we actually had 2 blooms occur again in this upper region of the pond. And then you can see how that kind of stacks up compared to the bloom that we saw last year. So 2022 actually had a more intense bloom than 2023, which is a good example of how things can change in the same system from year to year. Moving on to Squibnocket Pond, also up island, located here. This is also a brackish pond. We don't have a ton of environmental monitoring data from the main body of the pond. We have our one station here, which is kind of just adjacent to the public swimming beach, Squibnocket Beach. This pond does have fairly brackish conditions present, maybe a little lower on the brackish scale, depending on overwash influx and other factors. The patterns of cyanobacteria growth that occurred last summer followed a much more kind of standardized bell curve. It started low and then kind of slowly increased as the summer went on. Again, we are right in this kind of peak growth period between August and late October. It stayed high throughout that kind of peak time of high heat capacity in the water, and then kind of crashed as we got into the fall months. Taking a closer look at that bloom, it was characterized primarily by this genus Planktothrix and our old friend Dolichospermum, same as Chilmark Pond. The interesting thing about this particular bloom is you can see there's a lot of this algal material here, and this is actually a species of green algae, a harmless green algae, green filamentous algae actually, that was actually blooming at the same time. And what was happening was this algae was forming a medium that kind of captured and coalesced and condensed the existing cyanobacteria that was existing and growing in the ambient water column. And so if you were to take a sample for cyanobacteria out here, kind of beyond the algal mat, it would return a much lower concentration of cyanobacteria than it would right in the middle of this green algal bloom. So this is an important thing to recognize, in that sometimes the green algae and the cyanobacteria can kind of coincide together or compete together in the same space. And green algae can actually act as kind of like a net for concentrating cyanobacteria right in this interface zone. Then looking at Squibnocket over the years, so we first started monitoring in 2021 when reports of that large-scale bloom occurred in the same area. If you remember that aerial photo that we showed you at the beginning of the presentation, that was a much more diffuse bloom that occurred throughout the entire water column in that area. And you can see, compared to other years, it was a much higher, more intense bloom. Looking at 2022, it actually stayed fairly low throughout that season. And then 2023, the bloom we just analyzed in comparison to 2021 was kind of lower, but still in a bloom state. Moving over to West Tisbury and Tisbury Great Pond. And so Tisbury Great Pond had a bit of a unique case last summer, which I'll kind of elaborate on in a moment. But the results of our standard monitoring, so our standard water grabs and visiting all of our monitoring stations are presented here. And so Tisbury Great Pond is a brackish pond, again, on the south side of the island. It does have a decent amount of freshwater influx coming from two brooks and streams, the Millbrook here and the Tiasquam, entering into Town Cove. There's also groundwater input that comes in through the tips of the coves. So what you get in Tisbury Great Pond, it tends to be a bit more fresh conditions in the tips of these coves, as opposed to the southern portions of the ponds, which are more typically brackish. We also have Black Point Pond here that filters into Tisbury Great Pond through this narrow creek down here along the southern shoreline. And Tisbury Great Pond is also open to the ocean on a fairly regular basis throughout the year, supplying it with seawater and flushing the pond out. Looking at our cyanobacteria monitor, or standard monitoring data from throughout the season, you can see that Tisbury Great Pond remains fairly consistent. It has not a lot of variation between samples. Take note of the scale here. We're in a much lower scale. So overall, in the water column in Tisbury Great Pond, we were detecting lower concentrations of cyanobacteria. It never actually surpassed the alert status throughout the 2023 season, at least in the standard monitoring samples. Looking at Tisbury Great Pond over the last couple of years, 2021, we saw very low incidence of cyanobacteria activity. In 2022, slightly higher, but still, again, this low range below the 10 microgram per liter limit. And then also in 2023, a lot of similar things, and most of it occurring in that same time frame. Where things got interesting with Tisbury Great Pond was that the pond did actually experience a widespread cyanobacteria bloom, and it was of this particular genus, Aphanasthesae, which is kind of unique in its biology in that it forms these concentrated macroclusters of macro material or physical material that you can see with the naked eye. It seems to persist on the bottom or at deeper depth. So it kind of coats the bottom, or it can coat a shoreline along the pond. It was not actually visible until kind of mid-July when Tisbury Great Pond was open to the ocean and there was a rapid drop in water level, and suddenly a lot of this material was exposed. It looks a lot just like green algae, kind of this dark to bright green complexion. And if you were to take a water sample directly above this mass or even next to it, it would return a very low concentration of cyanobacteria. However, if you were to actually sample the physical material itself, it popped back very high. So what's different about this type of cyanobacteria is that it is very confined to the actual physical material itself, which is a bit confounding for our regular monitoring of just looking for diffuse cyanobacteria in the water column. So this was kind of a new case for us. We had some great response from the community. We had volunteers coming out to help us try to map the extent of the bloom. We were given the means to actually do some intensive surveying across the pond. And we did find that this material and this Aphanasthesae was at first appeared in Tyus Cove, and then it started to kind of bloom in the tips of the other coves throughout the pond into late August when it finally did die back. And so this is kind of something that evaded our normal monitoring at first, but now that we know kind of what to look for, we're kind of always on the lookout for it, and it's something to know going forward. Also in West Tisbury, we have James Pond. This is also a brackish pond. It has a thin connection to the ocean here on the northern end. It tends to be, in the southern end of the pond tends to be a bit more fresh than the northern end, a bit more brackish. We only have one monitoring station on this pond. It's kind of right here adjacent to the more public-facing areas. So Lambisco Beach is here, and also the land bank property, the new land bank property here. Looking at the trends from last year, again, we see kind of just a more normal bell curve of cyanobacterial growth. We did achieve a bloom status or a bloom advisory starting in early or late July. followed by kind of a typical crash that brought it down into lower levels for the remainder of the season. This bloom was characterized again by Dolichospermum, very common actor that we're starting to see. And again, it kind of it seemed to be mostly concentrated in the interface zone due to those wind-driven effects. So on the leeward or on the downwind side of the pond, we would see a lot more of this material kind of collecting along that shoreline, that interface zone. Again, if you were to take a sample out here in the main part of the water column, it would be a bit more diffuse and diluted. And this material that you're seeing here is actually more of that green algae also kind of coalescing at that site. Looking at the other previous years, we can see there is some differences, and it's a good example of how things can change from year to year. But in 2022, we had low levels kind of throughout the season, and that is contrasted by these higher levels experienced in 2023. Moving over to Seth's Pond, this is a freshwater pond off of Lamberts Cove Road in West Tisbury, the popular swimming beach right here where we have our station. Thankfully, Seth's Pond stayed fairly low throughout the season, which is great to see. And looking at data from 2022 as well, it followed the same pattern. It never, again, we're in a very low end of the concentration spectrum here, and it never really surpassed anything above a 2, which is great to see. And I hope it remains that way. Also in West Tisbury, we have Watcha Pond, another freshwater ecosystem. This does not have an intermittent opening to the ocean. It is remains primarily fresh unless it gets some kind of overwash event across the southern shoreline. And again, throughout the monitoring season, which is this, for us last year, it was this peak cyanobacteria growth season, it stayed fairly low throughout, which is, again, good. And then looking at 2022, where we do have some data and some overlap in the monitoring season, again, it also stayed fairly low throughout the summer. Edgartown Great Pond, another brackish pond, similar to Tisbury Great Pond. It is characterized by more freshwater conditions in the tips of the coves. We have a large spread of stations throughout this pond to capture a lot of this environmental variation. It does have an opening that occurs intermittently here on the south shore to recharge the seawater. And cyanobacteria concentrations and the community structure here is a little different from other ponds, which is interesting. We believe that this is actually a marine species of cyanobacteria based on some genetic testing that we were able to complete in 2022. And it does follow kind of a very gradual growth period, peaking in that peak growth season for cyanobacteria throughout. But it did not ever achieve the kind of bloom status. It just kind of got into fairly higher concentration before coming back down in the fall season. Looking at differences between years, Edgartown Great Pond has a pretty stark difference. In 2021, we actually saw virtually no cyanobacteria activity at all in that pond. And that was kind of coupled, or that was contrasted by 2022, where we started seeing kind of a similar trend to what we saw in 2023 of this bloom in the peak growing season. And most likely it's this marine species of cyanobacteria, which is different from other ponds. Just to the east, Krakatuxet Pond, again, a brackish pond, but it tends to be more fresh on the scale. We have our monitoring stations throughout this pond. It is connected to Edgartown Great Pond through this narrow sluiceway here. But they only kind of exchange in one direction depending on the water level in Edgartown Great Pond. The stations are fairly consistent throughout. We had kind of a peak bloom that was pretty consistent, but it did not achieve a bloom advisory status last summer. That's contrasted from different years where, in 2021, it kind of mirrored more closely to what we're seeing in 2023. But then in between 2022, we did have a fairly large bloom that occurred in this pond. And that can, again, just is a great example of how things can change in the same pond from year to year, depending on the conditions. And lastly, we started monitoring in the Mink Meadows Pond in Tisbury last summer due to the reports of a large spread bloom that was occurring throughout that pond. And kind of so you can see it started in early July and was already in a bloom status when we got our first sample and kind of continued on and persisted throughout the rest of the summer. And this is an area that we are now monitoring again for this season to try and pull up some more long-term trends. And that bloom was characterized by these two genus of Microcystis and Oscillatoria planktothrix. And again, it was mixed in with that green filamentous algae that tended to kind of hold it and concentrate it right at the sampling site, right at the interface zone. So that was a lot of information. Thank you for listening. So I just want to cap off with some practical tips for folks going ahead of what to do if we see a potential cyanobacteria bloom. So first and foremost, avoid contact. Most algae are harmless, but cyanobacteria can produce toxins. You know, if there's a visible scum or a mat present, try to avoid contact with the water. And obviously the most at-risk, typically the most at-risk populations or groups for cyanobacteria or harmful algal blooms are small children and pets, especially dogs. You know, they splash at the water line. Dogs will drink every amount of water they see in front of them at any time, and that's a great way to, you know, ingest cyanobacteria if it happens to be present. If you think you see a cyanobacteria bloom, you can report it to your local Board of Health department. And if you have the ability to take pictures or record the location, that is great information to help us kind of figure out what's going on. In case of emergency or exposure, you know, so if you or someone you know has been in contact with a potentially toxic bloom and are having these or adverse health symptoms, you know, obviously contact your physician immediately. Likewise, pet or livestock, you can contact a veterinarian immediately, and then please, you know, follow up with the Board of Health as soon as you can, so that we can start to look into it and see and prevent other people from interacting with it as well. If you are curious and would like to do more research and get more in depth into the life cycles of cyanobacteria, past trends, what causes cyanobacteria blooms, all of that good information, the MV-CYANO website, located at greatpondfoundation.org/MV-CYANO, has got a lot of great information available to you. The EPA has an excellent page dedicated to harmful algal blooms in cyanobacteria as well. Mass.gov, the Department of Public Health, also has a great page. And all of this information can be accessed from the MV-CYANO website. So I highly encourage you to go check it out. And of course, if you ever have a cyanobacteria related question, or you want to report a bloom, or just anything at all, feel free to email us at mvcyano@greatpondfoundation.org. So that is it. Thanks for hanging in. I hope everybody enjoyed that. Thanks to everybody for attending and the Boards of Health for being here. And we can now take some questions if we have them.
I have a question. This is Chris Murphy.
Hey, Chris.
Hi. If you could just go back to your earliest part of the presentation on Chilmark Pond.
Sure. Let's see here. How's that?
Well, you're getting there. Okay. Perfect. So yesterday I received, or this morning maybe, an updated yesterday report on the middle pond. And I have a little trouble calling what we're calling this, but there's Chilmark Pond, then there's the upper pond, and then there's maybe the upper pond. The two sites that you show in the Chilmark upper pond, it looks to me like whatever this cyano problem is in Chilmark Pond is its highest point of origin that we're seeing is up in that brook that I don't know what it's called, but the brook that goes into Tilton Cove, Millbrook or whatever, comes from Peaked Hill down.
Right.
If you're seeing that high on this chart, that would be whatever it is, CP number 2, upper pond.
Mhm.
And then at the mouth or in the middle of the pond, a high reading. And then yesterday's readings were high in the same area. And that's a freshwater pond. Are you doing testing for anything other than nitrogen in there?
Yes, so we do, and we actually, that's a great question, Chris. And we actually added a station here in the upper pond. You know,
Yep.
I do encounter different references to, you know, each pond as I go through Chilmark, but yeah, we have added a station here. We are monitoring not just for nitrogen, but phosphorus, you know, a full nutrient suite. And then we are also collecting all the environmental data that kind of goes along with that. So temperature, salinity, pH, turbidity levels, Secchi disk, all that good stuff. And we are trying to take our cyanobacteria and phytoplankton data that we get from the floor probe and match those trends in our yearly ecosystem monitoring reports with what we're seeing in the environment, if that answers your question.
It sort of does, and I thank you for increasing your looking at that upper pond and the upper pond. One of the things that just seems obvious to me is if you went up the brook to where it crosses under South Road, and again up the brook further where it crosses under Middle Road, those are two easy places to get water samples. You could learn a great deal about what's coming down that brook right there.
Yeah, and you actually, you will be excited to hear, Chris, that we actually did some preliminary sampling in those areas just this spring. And we're kind of hoping to kind of continue on with that investigation as the year goes on.
I mean, it looks like there's a big finger pointing up that brook that says this is where the problem's coming from. But sometimes looks are deceiving. So how do we prove it?
Yeah, that's a great point. And you're right, it's collecting more data. It's measuring the nutrients and expanding our search upland into the watershed.
Chris, that's a great question. You're leading us ahead to the next step. As David alluded, we've got some samples being processed at the Marine Biological Laboratory. So first we start in the water and then we work out to the watershed, understand what the health is, and then move outward. And we do that kind of in stages. We get so far, we understand what's going in, and then we ask the next question. We're at the question of starting to look at the stream, so please stay tuned. And we have a question from the chat. From one of your Board of Health folks. So Sarah asks, how do cyanobacteria reproduce? And cyanobacteria are singular-celled organisms, but they can be colonial, they can be together. So they reproduce asexually, and there's the cells divide. The adult cells divide, make more, and they can do it really fast. And because they're single cellular, when you have a bloom, it can happen really quickly over time. And so we're not having to wait for a long gestation period. Unfortunately, we're waiting just for the conditions to be appropriate, and then the cells go wild.
I saw a map. This is Francie Shaw. I'm in California, so I'm in a different time zone altogether. But we're on Chilmark Pond, on Quitza and Menemsha Pond, and there was a bloom or some sign of it in Lovey's Cove last summer. I saw in one of the Chilmark information things they were sending out. And you didn't show any testing for Menemsha or Quitza or Stonewall Pond there.
Yeah, and we actually did, and I think Anna McCaffrey actually went out herself and did a round of sampling, I think during that same time period, if I'm correct. And we didn't show that in this presentation to kind of cut down time, but they actually did not come across any high concentrations of cyanobacteria as part of that sampling effort. But that's absolutely a situation where you don't ignore a report of a potential bloom. We might keep an eye on that moving forward. And if it does elicit more activity this summer, we will perhaps start getting better at locating where that might occur and possibly forming some regular monitoring as necessary.
I thought it was interesting with Chris's question about the water coming in, because I was thinking of it sort of backwards. And from that, it also looks like it's where the water gets stuck, which is true of Lovey's Cove. It's like the dead water part of Quitza.
That is a fantastic observation and point. So that's actually, where the water kind of stops movement. That's referred to as water residence time. And so that's kind of the amount of time that a unit of water takes to move from point A to point B in the system is it's an indication of stagnation or just low movement, low flow. And cyanobacteria they actually, we talk about blooms and accelerated growth, but on the spectrum of other types of phytoplankton, it grows relatively slowly, even in a bloom state. And it likes water that doesn't move, because that means it's got time to do its thing. So you're absolutely right. It's those pockets where you see kind of low flow or slow moving water, or areas where it's not moving at all, tend to be a key factor sometimes in its growth.
Who is
Do you
talking
want to
about
stop
what we
sharing your
should do?
screen for a second?
Oh, sure.
And it looks like we have another question from Ed, or a new question from Ed McEntee. He says, how do these blooms affect shellfish growth and reproduction? And how long does it take for them to recover once the bloom subsides? David, you might know more than I do. When we reached out to Mass DMF and to shellfish folks when we were starting this program, there wasn't a great understanding about how shellfish sort of respond or accumulate in terms of accumulating cyanotoxins. What they were trying to figure out is where in the shellfish it would accumulate. And this is one of those emerging parts of science where there wasn't a great understanding. I think there's more understanding about other types of bacteria and how they maybe in more ocean or full salt environments impact health, because there's a longer term data set. But understanding cyanobacteria specifically, I think there's more questions than answers. And the way the local shellfish folks have responded is if there's a bloom, according to CYANO, they've generally recommended not fishing or shellfishing. And so the recommendations on CYANO incorporate information from Mass DMF, the local shellfish wardens and constables. But that's something that constantly needs to be updated as we get more information. David, did you have other thoughts on that?
Yeah, no, that's yeah, I agree with everything you just said. It is kind of an emerging field. There are a lot of scientists and a lot of researchers around the world kind of currently looking at this exact question. A couple of things to consider is that generally, when the conditions present are great for cyanobacteria growth, it's also typically good for the other types of bacteria that do close shellfishing. So you often will they'll kind of coincide. If you have high cyanobacteria, you're probably going to see high bacteria, and that's probably going to lead to a closure on that front as well. And there are lots of questions that come from that, whereas cyanobacteria tend to be more surface dwelling organisms, and shellfish tend to be more bottom dwelling organisms. So it's there's a lot of factors to kind of consider in that, but that is a very good question that's still being researched.
Is Chilmark, it's great that the town is doing this, but is there also, I'm sure, some group doing how we can stop this, help stop it? Because we seem to be the main reason it exists.
Yeah, cyanobacteria, I mentioned earlier in the presentation, they've been around for a long, long time. And, they're kind of doing their thing. They have, what photosynthetic organisms do. They've got carbon dioxide, they've got nitrogen, phosphorus, warm temperatures. They're going to photosynthesize, they're going to consume, they're going to produce oxygen. That's what they do. When you start to see them blooming more and more frequently and more persistently, that's it's a sign of imbalance. And that is.
Are you saying that's climate imbalance or nitrogen or other?
Probably, a combination of all those fun factors. So, excess nutrients entering the watershed as a result of human development or other activities, changes in the water flow patterns, as well as changes in rainfall patterns, changes in ambient temperatures increasing throughout the year, increase of storm events, high wind activity that can stir up bottom sediments and bring more nutrients back into play. A lot of these factors, as they increase, they are all just happen to be factors that cyanobacteria appreciate. Not as
and
much
as to who's addressing this, Chilmark Pond Foundation is dedicated to preserving and restoring Chilmark Pond, using science to do that. And so that we've been partnering with them, and as well as the MBL, the Marine Biological Laboratory in Woods Hole. And so that's figuring out where the nitrogen's coming from. It's figuring out how to increase circulation when possible. It's figuring the different ways to keep the pond healthy. As we know, the temperatures are going to increase and the storms are going to increase. So if you haven't visited their website, chilmarkpondfoundation.org is a great place to learn more.
Thanks.
Any other questions, either in the chat, or you're welcome to unmute yourself, turn on your camera, ask whatever works. You have Board of Health agents from across the island here with a whole lot of knowledge too. I don't know if any of you all want to chime in.
I think I put them all to sleep.
No.
let's just give them a shout out then. The health agents, year after year are busy with questions you wouldn't even know existed. And they, at the busiest time of the year, make time to review cyanobacteria data on a weekly basis, get the signage out and keep you guys safe. So we just wanted to give them a shout out. And I'm seeing some of their faces pop up. Yay! These are our heroes.
Thank you, Emily and David, for doing such a great work and for the shout out.
Great. Well, what do you say, Emily? If people are thoroughly enriched with knowledge, shall we move along?
Close it here. You wowed them with your slides. Well done. Unless Nancy's coming to ask a question. Nope. Nice to see you, Nancy. Great job, David. That was a lot of work. Now you can go to sleep.
Fantastic. And just to kind of remind everybody, this information is available on our website. We're going to make this presentation also available in a PDF format for download. So if you'd like to kind of review the slides, review the data, click on some of those links, you're more than welcome to do that once we get it up. And as always, if you have a question about cyanobacteria or anything, Great Pond related, you feel free to get in touch with us. We're happy to chat.
Thank you all.
Thanks, Nancy.
Thanks, guys.
Thank you.